Hello and good day, everyone. My name is Nur Adlina binti Zainal Ambidin, and currently I am a Master in Molecular student at IMU. Today, I will be presenting my research idea regarding a comparison study between ARMS PCR and NGS technique for the detection of EGFR exon 20 insertion mutations in non-small cell lung cancer. Before we start, I would like to elaborate the contents of this presentation, which includes the background study, literature reviews, research idea, research questions, research objective and hypothesis, significance of study, research methodology, and references. So what is lung cancer? It can be described as the presence of growing cancerous cells found primarily in the lungs region. It is one of the most frequent diagnosed cancer worldwide and has the second highest mortality rate with an estimation of 1 in 5 death rates among the population. As you can see from the figure on the right, highlighted in blue color, which represents lung cancer, it is more commonly diagnosed in the male gender in comparison to the female. This is because of tobacco smoking culture is one of the most significant factors for developing lung cancer. Recently, air pollution has also been concluded as a significant factor for developing lung cancer, as there is an alarming rate of non-smoker patients being diagnosed with lung cancer among the Asian geography as seen in the figure, especially among the females in China. But overall, the cause of high death rate in lung cancer is mostly due to the degree of malignancy, late detection, and diagnosis. Lung cancer can be categorized into two types, which is small cell lung carcinoma and non-small cell lung carcinoma. Non-small cell lung carcinoma is by far the most common subtype of lung cancer, being diagnosed with 85% of cases worldwide, and it can be further broken down into three categories, which is adenocarcinoma being the most common, followed by squamous cell carcinoma and large cell carcinoma. In the studies of non-small cell lung cancer, there are 6 common oncogenic drivers being identified, which are EGFR, KRAS, ALK, ROS1, BRAF, and MET. But today, primarily, we will focus on EGFR, which makes up of around 50% of non-small cell lung cancers worldwide, especially among the Asians. So, EGFR is a transmembrane protein receptor that is made up of 31 exons, and the component of the receptor itself consists of an extracellular ligand binding domain, a hydrophobic transmembrane region, an intracellular receptor tyrosine kinase domain, and a C-terminal domain. Activation of this receptor will lead to multiple downstream signaling, and the activation process of the receptor is through the mechanism of ligand-induced dimerization. You see, how it works is, when a ligand binds, it facilitates a conformational change to occur in the RTK domain for homodimerization or heterodimerization. The dimerization of the protein will actually lead to autophosphorylation of the tyrosine kinase residues found in the intracellular tail of EGFT, which will serve as an attachment site for other protein complexes, hence prompting downstream signaling pathways such as RAS, MAPK, PI3K, and STAT pathways. Mutations are most commonly found in the RTK domain of this EGFR gene, which actually makes the RTK domain itself as a key target for treatment in non-small cell lung cancer. Around 90% of the EGFR mutations are made up of classical sensitizing mutations, which are known as exon 90 deletions and exon 21 L858R. The other 10% consists of the uncommon EGFR mutations, which are known as exon 20 insertions, exon 21 L861Q, exon 18 G719X, and exon 20 S768I. Resistance to treatments among patients diagnosed with EGFR mutations are mostly due to the emergence of secondary mutations like exon 20 T790M and exon 20 C797S. EGFR exon 20 insertions are actually the third most common mutation found in the EGFR gene. It can be described as an in-frame insertions or duplications that are found at the position of 761 and 775 of the EGFR exon 20 gene. It is mostly heterogeneous at the molecular level, with each distinct variant showing different responses in treatment toward EGFR inhibitors, as stated by most studies. In EGFR exon 20 mutations, it is commonly found at the C-terminal end of the C-helix region. The C-helix actually plays a crucial role as a critical regulator that promotes the activation of EGFR and stabilizes the dimerization competent form of the receptor. But with the presence of EGFR exon 20 mutation, insertions are found at the phosphate binding loop region, resulting in a notable shift in the C-helix region, forcing it from an outward position into an inward position, which is the active state as seen on the diagram on the right. This actually causes activation of EGFR without any ligand binding, but it also does not affect the binding affinity the receptor has for ATP. This actually, however, results in a significant steric hindrance in the drug binding pocket and also resistance against first and second generation of EGFR inhibitors. Small molecule kinase inhibitors are the gold standards in treatments for non-small cell lung cancer, especially for classical EGFR mutants, because of its high sensitivity towards mutant EGFRs over wild type. However, the challenge in targeting exon 20 insertions for treatment is that not all variants of EGFR exon 20 insertions show an effective response towards first and second generation of EGFR inhibitors, such as the variant D770 here, which shows similar resistance mechanism as the T790M resistant gene mutation. It was later resolved by Yasuda in 2013 with his team that insertion in exon 20 that occurs within the C-helix region before residue position 764 shows a mechanism that is similar to those found in the classical EGFR mutants. Therefore, this type of variant, exon 20 insertion variants, actually shows high sensitivity towards first and second generation of EGFR inhibitors. And the good news is that in 2021, two targeted therapies has been approved by FDA for treatment of non-small cell lung cancer EGFR exon 20 insertion mutants, which are the small molecule kinase inhibitor mobocertinib and the anti-EGFR MET bispecific antibody amivantamab. Mobocertinib is actually an irreversible covalent kinase inhibitor with a similar structure to the third generation of EGFR inhibitor known as osimertinib. But the difference in structural is that mobocertinib actually has a C5-carboxyl isopropyl ester group in the middle of the pyrimidine ring. This actually allows it to be highly selective towards EGFR exon 20 insertion mutations, as it is able to exploit the selectivity in the narrow drug binding pocket that no drug is able to occupy for EGFR exon 20 insertion mutations. And this is actually demonstrated in the clinical trial for XClaim, where mobocertinib actually reported a progression-free survival of 7.3 months with tolerable toxicity profile among the patient cohorts, hence accelerating approval from the FDA for mobocertinib as a second-line treatment for non-small cell lung cancer EGFR exon 20 insertion patients. Amivantamab is another FDA-approved inhibitor demonstrating activity against exon 20 insertions. It is a human monoclonal immunoglobulin G subclass 1 based bispecific antibody that simultaneously targets EGFR and MAT pathways through 4 action mechanisms, and you can see them being described and summarized on the right diagram. The 4 activation mechanism is that amivantamab will bind to the EGFR receptors, thus blocking ligand-induced dimerization activation. Secondly is that the antibody-bound receptors will be internalized and degraded by the cell. The third mechanism is through the recruitment of macrophages, hence triggering antibody-dependent cellular trogocytosis. And the last mechanism is through the activation of natural killer cells, triggering ADCC, which overall both of these systems will actually lead to programmed cell death. Here are some of the literature reviews that has inspired this study. The first two describing the global trends and variation found towards lung cancer in addition to the epidemiology of lung cancer worldwide. It also highlighted how variation of lung cancer differs depending on the geography. The next few literatures are regarding the structure, variation, and mechanism of EGFR exon 20 insertion in lung cancer. It also highlighted the growing need for treatment solutions in regard to the different alteration variants found in exon 20 insertions. And lastly is the comprehensive analysis of NGS and ARMS PCR for detecting EGFR mutations by He and his team in 2022, which describes the available techniques in the market for detection of EGFR mutations in non-small cell lung cancer. This research idea is actually being inspired by the National Cancer Comprehensive Network, which has recommended the use of ARMS PCR or NGS technique for targeting gene mutations in non-small cell lung cancer before proceeding for treatment. And I actually hope that this experiment can be a landscape for future improvements in the detection of EGFR exon 20 insertion mutations, as well as assist clinicians in choosing which platform to be more suitable for detecting mutations in non-small cell lung cancer before proceeding for treatment. Amplification refractory mutation system PCR, or also known as ARMS PCR, is unlike traditional PCR because it is highly sensitive in detecting frequencies of gene alterations, as well as it requires a small amount of starting material, making it suitable for clinical diagnostic setting. It also has a good time efficiency as the results can be obtained quite rapidly, and it is a cost-effective technique because it can detect multiple targeted genes at the same time. But it also has its limitation, which is it is limited to the detection of known pre-specific gene mutations only. Next is next generation sequencing, which actually has a wide coverage for detecting gene mutations either known or novel. And it also has the ability to quantify tumor mutational burden and sequence of resistance gene mechanism. It is also a non-invasive technique and cost-effective as well because it can detect multiple genes at the same time. However, the limitation of this technique is actually it is quite time-consuming and tedious in the laboratory workflow. The research question for this experiment is that what are the most commonly variant found among patients diagnosed with non-small cell lung cancer EGFR exon 20 insertion mutations within the Malaysian population? And which is the most suitable platform for the detection of EGFR exon 20 insertion mutations in non-small cell lung cancer? Next is the research objectives. Consists of to identify the common variants of EGFR exon 20 insertions among the non-small cell lung cancer patient cohorts within the Malaysia's population. It is also to investigate the most suitable platform for the detection of EGFR exon 20 insertion in non-small cell lung cancer cases. The significance of this study is to identify the most common variant known among the non-small cell lung cancer EGFR exon 20 insertion patients within the Malaysia's population, thus encouraging the exploration of clinical trials available for further improvement in the treatment of EGFR exon 20 insertion cases. It is also to reduce the frequency of false negative result and misdiagnosis among the non-small cell lung cancer patient cohorts. The null hypothesis for this research is actually the NGS platform being the best platform used for detecting known and novel EGFR exon 20 insertion mutations due to its high sensitivity and specificity along with a broader coverage across the EGFR exon 20 gene. This is an initial overview methodology plan for the research idea where there is a sectioning of FFPE or cell blocks for patients with known diagnosis of EGFR exon 20 insertions, followed by the process of deparaffинization of tissue sections and extraction of DNA and RNA simultaneously. The extracted nucleic acids will then be quantified by using qubit or nanodrop respectively before proceeding for ARMS PCR and NGS workflow. The final stop will then be analysis of data from both techniques for a comparison study. Hence, we have reached the final slide for the presentation, which is the references for my slides. Thank you for your time today and goodbye.